Hello, class. I'm going to go through the eye and ear models and dissection. Here is a picture of our ear model that we have in our lab. This portion over here right here is the pinna, also known as the auricle. This region here, this tube, is called the external auditory meatus, also known as the auditory canal. Do not confuse that with this portion here called the auditory tube, also known as the eustachian tube, also known as the pharyngotympanic tube. It connects your throat to the middle of your ear. There are three bones that are your middle ear ossicles. On another slide, these will be more clear. The ossicle attached to this structure here, which is called the tympanic membrane or tympanum, the ossicle that attaches to that is the malleus. The malleus articulates in turn with the incus, and then the incus articulates with the stapes, which basically vibrates against the oval window. We'll see that soon. These bony structures that look like arches are the semicircular canals, and there are soft membranous structures inside of them called the semicircular ducts. This structure over here that looks like a snail shell is the cochlea. So here is the same model, but with a more superior view. Again, here would be the tympanum, and you can see the malleus articulates with it. Then we have it articulating with the incus, which articulates with the stapes. There is a muscle right here called the tensor tympani muscle. You will learn more about that muscle when you learn about your cranial nerves. Over here, we again see the snail shell called the cochlea, and the nerves that are coming off of it are going to comprise the vestibular cochlear nerve. There is a vestibular branch that innervates basically the vestibule and the semicircular canals, and there's a cochlear division which innervates the cochlea. Together, those create cranial nerve number 8. Here is another ear model that we have in our lab, and all of the structures are identified here. Then again, we have the pinna, we have the external auditory meatus for number 2, we have the auditory tube for number 3, number 4 is the malleus, again, which attaches to the tympanum right here. We have a semicircular canal and ducts, so I need to fix this. That should not read semicircular canal, that should read semicircular duct. And that's what I'm pointing to here with the blue arrow. The semicircular ducts are the soft membranous structures within the semicircular canals. Again, with number 9, we see the vestibular cochlear nerve. Number 8, we see the cochlea. This structure right here, hidden, is the vestibule. Inside the vestibule, we would find the utricle and the saccule. These swollen endings of the bony semicircular canal are called ampullae, ampulla for singular. Inside the ampulla, within the semicircular duct, we would find the crista ampullaris. The semicircular ducts embedded within the semicircular canals, the bony structure, they are important for helping your brain interpret rotational movement. The vestibule with the utricle and saccule, they give information to the brain through cranial nerve 8 about the placement of your head. Is it resting on your chest? Are you looking all the way up? How fast are you accelerating? So that's the information that comes from the vestibule. The information that is sent to the brain from the cochlea will be about sound, your hearing. Here is a more up-close image of the semicircular canals. Again, the soft membranous structure that's inside, that I'm highlighting here, is the semicircular duct. Here, for letter C, we're seeing the oval window, and that is where the stapes would be pushing on. Over here in the background, we're seeing the vestibular portion of cranial nerve number 10. This big region here, all of this region here, is the vestibule. And again, inside of it would be the utricle and saccule. And then over here, we're seeing the cochlea, the snail shell again. Here's another view of this. Here are our semicircular canals. And over here, we're seeing inside of them the membranous labyrinth, the semicircular ducts. This blue-looking structure is a semicircular duct that has been removed from one of these semicircular canals, just so you can see the soft membranous structure. Again, down here, we're seeing a window. In this case, this is the round window. And then over here, we're seeing the cochlear nerve coming off of the cochlea. Together, the vestibular nerve and the cochlear nerve create cranial nerve number 8, vestibular cochlear. Here is another picture showing you the ossicles. These are the ossicles. Here's another ossicle. And here is the tympanum or tympanic membrane. Again, we can see the semicircular canals with the soft membranous structure inside the semicircular ducts. Letter M down here is the stapes. Letter J up here is the incus. And this bone that's attached to the tympanum is the malleus. Again, we can see the cochlea, and letter N is pointing to the vestibular portion, and O is pointing to the cochlear portion of cranial nerve number 8. Yet again, another picture. We see the semicircular canals. This is the lateral. This one up here is the anterior, and that one in the back is the posterior semicircular canal. The soft membranous structures inside are the semicircular ducts. Down here is the vestibule, and inside we can see the utricle and saccule. I always remember S for saccule and S for small. It's the smaller of the two membranous labyrinths within the vestibule. Utricle, saccule. Again, we see the cochlea. Okay, continuing with our ear and eye demonstrations for lab. Here is a picture of the cochlea basically cut through. We see two spaces here. We see the scala vestibuli, and then down here is the scala tympani. These would be filled with perilymph. These are the bony containers, and in between the two is the soft membranous structure called the cochlear duct. This membrane up here is the vestibular membrane, and down here is the basilar membrane. We can see hair cells right here. They are part of the organ of Corti, and in another picture we'll take a more up-close view of this. Here is the cochlear nerve and the cell bodies, the ganglia cells. Again, this would be a component of cranial nerve 8. Excuse me. This structure right here is the tectorial membrane. Here's a more up-close picture of the organ of Corti, circled here. Again, the vestibular membrane we see. Down here is the basilar membrane. Over here is the scala vestibuli and scala tympani. Again, those structures would be filled with perilymph. The soft membranous structure found between them is the cochlear duct, also known as the scala media. It would be filled with endolymph. Again, we can see the endings, the nerve endings of the cochlear nerve. We can see the hair cells here, and we can see that the cilia of those hair cells are touching the tectorial membrane. Here is a non-labeled view of the cochlea. Can you identify the structures 1 through 8? So number 1 would be the scala vestibuli. Number 2 would be scala tympani. 4 is the vestibular membrane. 5 is the basilar membrane. 3 is showing you the cochlear duct or scala media. 7 is showing you the tectorial membrane. And 8 is basically pointing to the hair cells. Altogether, the tectorial membrane, the hair cells, and the basilar membrane create the spiral organ of Corti. Now we're moving on to the eye. We can see with letter A, I'll go in order, is the optic nerve. Letter B is pointing to the superior rectus. Letter C is pointing to the superior oblique as it turns through a ligament called the trochlear ligament, shown here in letter D. Over here, this muscle is the lateral rectus. And we're looking deep inside the orbit here. In the background, we can see the medial rectus. G is pointing to the lacrimal sac. The lacrimal gland makes eye secretions, your tears, and they are swept medially, and then eventually those tears, those eye secretions, end up in the lacrimal sac. Letter H is showing you the iris. Letters I and K are pointing to the two different kinds of smooth muscle that comprise the iris. There's a radial muscle that radiates out like the spokes of a bike, and then we have sphincter muscles. Those two types of smooth muscle would control the diameter of the pupil, shown here, this hole. If the radial fibers contract more, you have pupillary dilation. If the sphincter muscles contract more, you would have pupillary constriction. And lastly, down here, we see the inferior oblique. Here is another picture of an eye model that we have. This clear region right here, outer region, is the cornea. This white portion here on the sides, the white of your eyes, is the sclera. Together, the cornea and sclera create your fibrous tunic. Again, the hole in the middle is the pupil, and this structure here, the colored part of your eye, is the iris. The smooth muscle fibers in the iris control your pupillary diameter. Back here, this colored or pigment part is called the choroid. There are three parts to the vascular layer or vascular tunic: the iris, choroid, and you will see in another picture the ciliary body. Here is one of our lab models taken apart so we can see a side view of the eyeball. Again, the hole in the middle here would be the pupil. This part right here going around it, that is your iris. Then back here we had the ciliary body. Back here, it's not really showing up very well, is it? Ciliary body, which looks like basically the underside of a mushroom cap. This wavy part in the back here is called the ora serrata, and again, you should add that to your lab hit list. The region behind the iris is called the posterior cavity, and this would be filled with vitreous humor. This orange substance here is showing you the retina. The retina is part of the neural tunic. The vascular tunic, just to say it again, has three parts: the iris, ciliary body, and this colored region down here, the choroid layer. Over here, this is called your blind spot. Technically, properly, it's called the optic disc, and this is where axons are exiting your eye from the retina. There are no photoreceptors there, which means that region cannot detect light or color, and that's why it's called your blind spot. Here is another eye model. I will go through them one by one. The white portion out here is the sclera. Deep to it, the colored portion is the choroid, part of the vascular tunic. And then the yellow portion shown here is the retina, part of the neural tunic. The fibrous tunic is comprised of the sclera, shown here with number 1, but also the cornea, shown here as number 4. Number 5 is the iris, part of the vascular tunic. Number 6 is the hole going through the middle of the iris called the pupil. Number 7 is showing you suspensory ligaments that come off of, shown down here in 7b, the ciliary body. Number 8, we're seeing the lens. And number 10, shown back here, number 10 is pointing to a chamber behind the iris, posterior to the iris, but anterior of the lens. So number 10 is pointing to the posterior chamber of the anterior cavity. Number 9 is a chamber found behind the cornea, but in front of the iris. So number 9 is pointing to the anterior chamber of the anterior cavity. Both these chambers, anterior and posterior, are part of the anterior cavity. That's number 11. And would be filled with aqueous humor. Number 12, this whole region back here, this region is the posterior cavity, which would be filled with vitreous humor. Number 13 is showing you the fovea centralis. This is where you have the most dense population of photoreceptors and gives you your best visual acuity. Number 15 is showing you the axons exiting out of the eyeball and the optic nerve, and this region is your blind spot, most properly known as your optic disc. So here it is again, just a more up-close picture, and it's labeled all the same way as it was in the previous. We have our chambers of our anterior cavity. So here's your anterior chamber, and behind the iris, but in front of the lens, would be the posterior chamber. Here's another view of an eye model that we have. We can see the sclera, but the more superficial tissue that would be on top of the sclera is the conjunctiva. The colored portion that you can just see a little bit here is the iris. Excuse me. Here would be the whole of the pupil. We can see the suspensory ligaments coming off of the ciliary body. Here's the ciliary body. And as we learned in the previous slide, the anterior chamber of the anterior cavity would be filled with aqueous humor. It is actually the ciliary body that makes that. The aqueous humor comes from the ciliary body, goes into the posterior chamber of the anterior cavity, then comes out through the pupil into the anterior chamber of the anterior cavity, and then drains back into your plasma through the scleral venous sinus, also known as the canal of Schlemm. Out here in the dark color, we have the choroid. This yellow region, part of the posterior cavity filled with vitreous humor, is where we would find the retina, part of the neural tunic. So here is the same model again, but from a more anterior perspective. Again, we can see the colored iris, the hole in the middle is the pupil. You can see the choroid. The orange region represents the retina. Where the axons are exiting is the region called the optic disc, also known as your blind spot. The area shown here on the back of the retina is the fovea centralis. Another model in our lab shows the fovea centralis as a purple dot at the back of the eye. There is a region around it called the macula lutea, and this is where we start to get less populated photoreceptors, at least compared to the fovea centralis. Now, this is a cross-section of the sheep eye that has been dissected, and I'm going to show you another perspective of this. And in our YouTube videos, I actually showed you how I did the sheep eye dissection. The dark substance here that is revealed once I scraped away the retina is the choroid. Animals have this shiny region called the tapetum lucidum. It allows animals that have this to kind of reflect light. Maybe you've driven home and you've seen a cat out on the street, and when your headlights hit it, it looked like their eyes were glowing. That's because of this kind of fluorescent looking substance called the tapetum lucidum. Again, the mushroom cap, kind of difficult to see in this picture, is the ciliary body. And then we have the iris. We have the anterior chamber of the anterior cavity. Here's the lens behind the iris, but in front of the lens would be the posterior chamber of the anterior cavity. This whole region back here is called the posterior cavity, and it would be filled with vitreous humor. So here's another perspective of a dissected sheep eye. It's easier to see the mushroom cap appearance of the ciliary body. The flat region here is the iris. In my dissection video, it was very difficult to show you that, but I did try. We can see the cornea through the pupil here, so that would be the anterior portion of the eye. Here we see the lens, and again we see a portion of the ciliary body. And all of this jelly fluid here is the vitreous humor. Out here, the white of the eye is the sclera. So this is the sheep eye dissection where I opened it up Again, you can see the jelly substance, the vitreous humor. This region right here is the lens. The yellow portion here is the retina. And actually right there, you can see where the axons are going through the optic disc. Deep to the retina is the black substance called choroid.